bar.
All right, so our second presenter today is Dr. Ava Quijano Cardé. She is a third-year microbiology PhD student at the Aquatic Animal Health Lab at UC Davis under the guidance of Dr. Esteban Soto. Her professional interests focus on fish virology and fish veterinary medicine. In her free time, she enjoys quality time with friends and family, scuba diving, and salsa dancing. And today, Ava is going to be talking to us about the whole genome sequence of acid-penserid herpes virus 2 and the identification of potential host homologs. So go ahead, Ava.
Thanks, Megan. And thanks everyone for joining me today. I also wanted to thank the organizers of the seminar series because this is a great opportunity to share science and learn from others. So I'm pretty happy about it. As Megan said, my name is Ava Quijano Cardé. I am a now third-year PhD student at UC Davis under the guidance of Dr. Esteban Soto. And I'm going to talk to you a little bit about my journey sequencing and assembling the whole genome of acid-penserid herpes virus 2, and a little bit on the initial identification of potential homologs in that genome. And I just want to take a moment to thank all my outstanding co-authors for making this work possible. Many of you are here today. There we go. So first, let me introduce to you the amazing white sturgeon. These fish are native to the west coast of North America and have remained basically unchanged since the early Jurassic period, making them, in essence, living dinosaurs. And you can get an idea from that by the way they look. They look a lot like sharks. They're also the most important species of sturgeon in the United States aquaculture industry, being responsible for approximately 95% of the caviar and sturgeon meat production. So this captive fish multi-million-dollar operation, which is primarily centered in California and the neighboring Western states, generates outstanding revenue for the producers and local employment opportunities, but it also indirectly protects our wild populations from overfishing for consumption. An important infectious pathogen of white sturgeon is acid-penserid herpes virus 2, or ACHV2 for short. Those of you that have been in the field for a little bit longer or work with these type of viruses may have known it as white sturgeon herpes virus 2. This is a large double-stranded DNA virus in the family Alloherpesviridae that causes a variety of clinical signs, including this pretty extensive erythema and epidermal ulceration that you can see in the images from our experimental cases. Mortality of up to 10% in subadults and 50% in juveniles has been documented in natural outbreaks, with even higher mortality rates in laboratory control studies, depending, of course, on the infection doses. So besides these very significant clinical signs as the herpes virus, it is suspected that ACHV2 has a potential to establish latency. And we have seen devastating subsequent outbreaks in our survivor fish from our challenges after the challenge has culminated. Oops, sorry. So one of the big limitations for the pathophysiology research that we want to do on the potential latency of ACHV2 is the lack of a complete genome for ACHV2. So we decided to sequence 4 isolates of ACHV2 from natural outbreaks in the Western United States throughout the last 2 decades, and those are the locations where those isolates were originally isolated from. I cultured each isolate in white sturgeon skin cells at 20 degrees for 7 days and monitored for cytopathic effects. And here at the top is how the WSSK cells, healthy and happy, look like. And then at the bottom, you can see the characteristic cytopathic effects, where you see rounding of infected cells and formation of syncytia with detachment of severely affected cells. So at that point, I collected the virus, extracted DNA, and amplified it to increase the low viral DNA that you usually find in viral culture. I then sent the DNA samples for both Illumina and nanopore sequencing. The Illumina sequencing was done by GeneWiz, which is known, I think, now as Ascenta Technologies. And then nanopore sequencing was done by Matt Griffin's lab, which is one of our outstanding collaborators that you all know. Dr. Titus Brown and his lab were instrumental in helping me figure out how to do all this coding and obtain the context for the Illumina reads, so thank you. I started with some Kmart Exploration, which allows you to assess the raw data and estimate the genome size. So for those of you that may not know Kmart, these are overlapping genome fragments of a predetermined size that facilitate immensely the processing of the data and significantly reduce the computational time. So in this example, for example, you would have 7 mers because they're Kmers of 7 nucleotides. And we have evidence that at the 100 kilobase level, which is where we expect CHV2 to be, the error in estimation is less than 0.02%. And this is what Kmart Exploration looks like. So your genome of interest will form this bell curve right here when you sort the Kmers, and then you can use the second column to estimate genome size. And with the estimated genome size, we calculated coverage, which speaks to the number of reads that align to a reference sequence. So in general, the higher the coverage, the higher confidence with which you can determine each base. But it's been shown that if you have excessive coverage, this can lead to long computational times, and for some of the errors to be called as non-errors because the assembler will encounter them time and time again. So for the Illumina sequencing, we obtained a 6,000x coverage, which is outstanding. And we decided then to subsample the reads to have a 250x coverage. The recommendation for a good whole genome assembly is around 45 to 100x. But since our genome size, it was an estimation, we decided to err on the side of caution and stick to 250, which is still much more reasonable than 6,000. After assembling the Illumina context, I noticed that the resultant genome size was significantly higher during quality assessment than the estimated one. And this is usually an indication of contamination, which we suspected to be from the WSSK cells that we use for propagation. And this is actually pretty common for viral culture. So at the time that this analysis was being done, the white sturgeon genome was not available, which it is now. So I employed Kmart frequency clustering to filter out the context with lower abundance as contaminants. So here's an example of what that looks like. We can see here at the top that 53,243 Kmers repeat only 120 times. So these are considered to have low abundance and were filtered out. So what is the disadvantage of Illumina sequencing? So imagine you have this genome here at the top with tandem repeats. When you do Illumina sequencing, you obtain all of these short sequences that overlap. But when you give the assembler these short reads, it has a really hard time deciding if there's a bunch of repeats, like there are, or if there's only a handful of repeats. So quite often what ends up happening is that you get the incorrect number of repeats and deeper coverage as the reads just stack on top of each other, that you can see on this image. This is where the nanopore truly shines. So the nanopore sequencing generates long reads capable of spanning the whole region of repeats and giving you the correct number of repeats in that region. So the context from our nanopore reads were assembled by my esteemed collaborator, Dr. Geoffrey Waldbieser, who is also here today. And in summary, he filtered the nanopore reads by quality and a minimum length of 1,000 base pairs to obtain approximately 90x coverage, which is excellent. After a single large context where each isolate was assembled, the nanopore reads were aligned to the nanopore context, and one wide round of base correction was performed. Now, this seems like a pretty straightforward assembly, the nanopore one, with a clear advantage. So why did we even bother to do Illumina? So based on this study by Delahaye in 2021, with the current technology that we have and the most mature base caller, the current mean error rate on high quality raw reads seems to be around 6%. And this is true for both prokaryotes here on the top and then eukaryotes here at the bottom. So taking that into consideration, we aligned the Illumina reads to the nanopore context and did another round of base correction on that assembly. So doing both Illumina and nanopore sequencing allows for best of both worlds and the most accurate assembly you can get at this point. But even with the best of both worlds, machine still needs a woman. So this is an image mapping both the nanopore and Illumina reads on the bottom to the nanopore contact on the yellow box. And on the top in blue, you can see the coverage obtained from this mapping. So generally, you expect that the coverage in a genome will be the same throughout the whole genome approximately. But if you direct your eyes right here, you can see that this region has about double the coverage, and that's an important thing to notice. So to help understand a little bit, I'm going to explain this concept. We know from studies in other herpes viruses, including ectolyrid herpes virus 1, which is the representative strain of the genus, and we heard a little bit about this virus last week. We know that herpes viruses circularize during replication. The mechanism for circularization is still under debate, but for the most part, people can agree that it's either formed by homologous recombination of the terminal repeats, forming an episome with a single terminal repeat copy, or by end-to-end ligation, forming an episome with 2 copies of terminal repeat. A fair number of studies, if you read a little bit about this, support the end-to-end ligation, which would explain how those herpes viruses that don't have terminal repeats still circularize. But those same studies acknowledge the supporting evidence for the presence of a single terminal repeat copy in most herpes viral episomes that do have terminal repeats. So I think the preferred hypothesis at this point is that it is end-to-end ligation with subsequent intramolecular homologous recombination of the terminal repeats. So the most likely scenario in our case is that our sample contained both episomes of ACHV2 as well as linear genomes. And if that's the case, then you can imagine that if a read is being generated from a linear genome close to the end, let's say right there, then your read would end where the linear genome ends, right there. If on the other hand, your read is being generated from a circularized genome near the end of the terminal repeat, somewhere around there, then your read could originate on the unique region at the end of the unique region and then continue all the way to the beginning of your unique region. And then the presence of those 2 types of reads is really problematic for the assembler because it will struggle to determine what is the true beginning and end of the linear DNA as it stacks the reads. So we suspect that it ended up placing all the overlap terminal repeats in this area of increased coverage and doing the best it could to determine the beginning and end of the linear DNA. So to help elucidate the correct beginning and end of our linear DNA manually, I generated this extended preliminary genome by aligning the context to the published partial ACHV2 genome. And this extended preliminary genome was then aligned to ectolyrid herpes virus 1, which is that second one at the top. And we can see that the extended one aligns really well to ectolyrid herpes virus 1 with some extra sequence at both ends. And I'm just going to put some boxes on those for you. So if circularization occurs, then we expect that this extra sequence at the beginning will be present at the end of the aligned region, something like that. And then the extra sequence at the end would be present at the beginning of the aligned region, which is exactly our case, as you can see here. A second supporting piece of evidence for the basis that I selected as correct beginning and end of the genome is that now we were able to identify long direct terminal repeats, which are the identical sequences of the 5' and 3' ends of the unique region, which we've been talking about. And I'll show you those in a second. The third piece of supportive evidence for the selected beginning and end of the linear DNA would be the presence of a large majority of nanopore reads that end exactly at those sites, all of these guys that you can see right here. So one final interesting thing during the manual correction was evidence that for ACHV2, there seems to be just one copy of the terminal repeat in the episomal form. And if you look at the reads that continue, whoops, yes, that one. If you look at these reads that continue past the beginning of the linear DNA, that sequence there corresponds to the end of the unique region right there. So homologous recombination of the terminal repeats is probably something that is happening in ACHV2. So after all that, I'm very excited to present to you the complete genome of ASCHV2. Each of these black lines is one of our 4 isolates, and the bar at the top refers to the similarity of bases within the alignment. So the greener, the more identity matches for each aligned base. Our isolates are approximately 134 kilobases in size, so very similar to ectodermal herpes virus 1, with 102 predicted open reading frames shared among them here in pink. 2 of those predicted open reading frames are the putative terminase and DNA polymerase. So the spliced, they are spliced, and the exons here are in red for you to see. In addition, each isolate has 1 to 3 unique predicted open reading frames, which we don't know the significance of those just yet. The alignment of these genomes reveals that over 98.6% of the bases are identical between any 2 isolates. Finally, as I mentioned before, the long terminal repeats in ASCHV2 are in blue here, and they are 1,907 base pairs with very little variation among the isolates. We're working on prediction of the identity of each ORF to be able to build a more robust phylogenetic tree. Although, to be honest with you, a lot of them are hypothetical proteins that haven't been explored more. And that's something that we hope to change in the upcoming years in this line of research. But similarity matrix built on sour mash confirms that these clustered together with the previous published partial genome of ASCHV2, and those are the orange arms of the tree, and separate from other herpes viruses of fish, which are the green and the blue arms on this tree. So switching gears somewhat, it's well established, particularly in mammals, that herpes viruses have multiple ways of regulating host cellular immune competency, resulting in immune system evasion. Unfortunately, few of these types of studies have been done in viruses that affect fish, but previous reports in Cyprian herpes virus 3 have identified viral homologs of IL-10 and the tumor necrosis factor receptors, which are important players of the immune response. I am particularly interested in looking into host homologs expressed during latency, because latency is a key evolutionary strategy of herpes viruses, and it's what makes them special. And it has allowed them to establish lifelong infections in their host via immunomodulation. And immunomodulation is pretty important because it can result in infection facilitation for other pathogens. And it's something that we suspect may be happening in the white sturgeon. The emerging infectious disease in white sturgeon caused by the gram-positive zoonotic bacteria called Streptococcus neae is associated with acute skin ulceration, body arching, and abdominal distension, which you can see particularly on this first image here from a clinical case, and can result in mortalities of 30 to 50% in subadult white sturgeon, which is incredibly detrimental for the aquaculture industry. We have studies that have shown that high doses of intramuscularly administered strep neae following a stress netting event are needed to induce disease in laboratory control challenges, supporting previous reports that have suggested that immunomodulation by stressful events such as transport or temperature fluctuations in aquaculture facilities may be responsible for infection facilitation in these cases. But what about those cases with no history of extraordinary stress? Knowing that subclinical ASCHV2 infection has been documented in broodstock populations, could the latent ASCHV2 be playing a role? And since 2015, multiple cases of co-infections of ASCHV2 and strep neae have been diagnosed in outbreaks in culture sturgeon here in Northern California. We, in fact, have preliminary data. That is the paper I was just talking about. But we have this preliminary data to show that a previous potentially latent ASCHV2 infection leads to higher mortalities during a strep neae challenge in juvenile white sturgeon. And you can see that in this mortality curve where the dark blue is the co-infection group compared to the single pathogens up there and the negative control. In similar cases to the white sturgeon co-infection, multiple reports of human streptococcus infections in the face of an already established latent human herpes virus 3 infection have been described and usually result in pretty severe disease in children. So these virus host bacteria interactions might span across taxa and are quite important to elucidate. And we have a pretty natural model here to study that via comparative medicine. So to screen potential homologs of white sturgeon in the ACHV2 genome, I used tBLAST-N to query my protein sequences to the recently published white sturgeon nucleotide sequence database, which is pretty exciting that it's out there in the world now. And when looking at protein comparisons that had an e-value of 10 to the minus 10 or less, which is considered statistically significant for homology at the protein level, 5 predicted proteins were a good match, but only 4 of them were present in all 4 isolates, which are the ones you can see on this table. 3 of them had putative conserved domains consistent with a glycoprotein and 2 protein kinases, while the 4th one had no conserved domain identified and no homologous characterized protein as a match, but it is homologous with hypothetical proteins identified in other members of the Alloherpes viridae family. So we still need to do quite a bit of work to investigate these open reading frames in detail, but one interesting case is the predicted glycoprotein, which in other viruses has been described moonlighting in immunovation and immunomodulation, both as an anchored and secreted protein. And there's that review, The Secret Life of Viral Entry Glycoproteins, is really interesting if anyone is quite interested in immunomodulation. So that's all the time I have, but I want to thank all of my co-authors and collaborators. This wouldn't have been possible without you. And I want to thank my funding sources at the NIH and the USDA NIFA for making this work possible. And we have very cool next steps going on in the lab already. So we shall see you at the next summer series with an update. I'd be happy to take any questions that people have now.
Thanks, Ava. That was a great talk. Very exciting work. Yeah, if anyone has any questions, go ahead and unmute yourself or drop them in the chat.
Let me open the chat here. More than assistance from Joff, Matt.
Any questions? We have several minutes still. Go ahead, Jim.
Eva Marie, that was really great. Thank you very much. I enjoyed that. Do you think are these immunomodulating proteins expressed during the latent phase at the same rate that they are during the active replication phase? And if so, is there some shift between those phases as far as the co-infection studies?
Yeah, great question. We don't know yet. That's the third part of my thesis. We are working on assessing the gene expression during the lytic cycle. And we're hoping to build a tool to be able to identify the tissue where the potential latent virus is. And once we do that, we will be able to do gene expression on those tissues when we expect the virus to be latent and see if there is a difference. By definition, right in the latent state, you have incredibly diminished gene expression at the cost of lack of viral production. And so we do expect those proteins to be expressed in decreased amounts, but sometimes you do have a shift between like genes that are turned completely off in lytic cycle and then they're on in the latent cycle. So you could still see like an increase. So that is going to be super interesting to see. And I'm very fascinated by what is latency and what you can see in those cycles.
All right,
But
great, thank you.
hopefully we'll have more for you soon.
I'm excited. Thank you.
Yeah.
Any more questions?
And peeps can always email me if they come up with questions later. I just put my email in the chat.
Sounds good. Thanks.
Thanks, everyone.
All right.
Oh, Dr.
Yeah.
Festival.
So yeah, thank